In this video, we are going to be discussing bone formation. So the process by which bone forms is called ossification. It's also called osteogenesis. And bone formation occurs in four principal situations. The first one is going to be initial formation of bones in an embryo or a fetus. Then the second one will be the growth of bones during infancy, childhood, and adolescence until adult sizes are reached. Third one is going to be remodeling of bone. So this is the replacement of old bone by new bone throughout life. And then the fourth one is going to be the repair of fractures throughout life. We'll first be looking at bone formation in the embryo. So the embryonic skeleton is initially made up of mesenchymal cells, and they are made kind of in the general shape of a skeleton, and this is the site where cartilage formation and ossification occurs. This is usually during the 6th week of embryonic development. So this is when 6th week of development is when we start to have bone formation. So there are two patterns of bone formation, both of which involve the replacement of a pre-existing connective tissue with bone and do not lead to differences in the structure of mature bones, but these are simply different methods of bone development. In the first type of ossification, which is intramembranous, this is where bones form directly within the mesenchyme. So this is in the mesenchyme, which is arranged, the mesenchyme are arranged in sheet-like layers that resemble membranes. In endochondral ossification, which is going to be the second one we will discuss, this is where cartilage is initially first formed in the correct shapes, and then it's replaced with bone. So we're going to have cartilage, replaced with bone. Okay, so here we have intramembranous ossification, and this is the simpler of the two methods. And the flat bones of the skull, most of the facial bones, the mandible, which is the lower jawbone, and also the medial part of the clavicle, or the collarbone, are formed in this way. So intramembranous ossification occurs in 4 steps. The first step we have here is development of an ossification center. So that is step 1. So at the site where the bone will develop, you have special or very specific chemical messages that cause the cells of the mesenchyme to cluster together and to differentiate first into osteoprogenitor cells and then into osteoblasts. And the site of such a cluster is called an ossification center. So this is going to be the initial cluster of mesenchyme cells. And this is where osteoblasts secrete the organic extracellular matrix of bone until they are completely surrounded by it. Step 2 is going to be calcification. So next, the secretion of extracellular matrix stops, and the cells, now they are called osteocytes, right? Because they are completely encased in the extracellular matrix that they were secreting. These osteocytes lie in a lacuna and extend their narrow cytoplasmic processes or extensions into the canaliculi. And these radiate in all directions. calcium and other mineral salts are deposited within a few days, and the extracellular matrix hardens. So here we have this bone extracellular matrix, and it becomes quite hard. Third step is going to be formation of the trabeculae. So as the bone extracellular matrix forms, it develops into trabeculae that fuse with one another to form spongy bone. This is around a network of blood vessels in the tissue. So you can see the blood vessels, and the trabeculae are actually forming around the blood vessels. Connective tissue associated with the blood vessels in trabeculae differentiates into red bone marrow. So this is also going to be the step where we have red bone marrow developed. Okay, then the fourth step is going to be development of the periosteum. So in conjunction with the formation of trabeculae, so this happens at the same time as the formation of trabeculae, the mesenchyme condenses at the peripheries of the bone, and it develops into the periosteum. So here, actually in step 3, you can already see this mesenchyme is condensing on the periphery of the bones, which develops into the periosteum in step 4. Eventually, a thin layer of compact bone will actually replace the surface layers of spongy bone. So you can see here, this compact bone. And the spongy bone, though, still remains in the center. So you have compact bone on either side, spongy bone in the center, and periosteum that is just outside of the compact bone. So even after this type of bone formation is completed, much of the bone will still be remodeled, which is going to be destroyed and reformed as the bone is transformed into its adult size and shape. So we could still have remodeling still occurs after. In endochondral ossification, bone forms with hyaline cartilage that develops from mesenchyme. Essentially, cartilage is formed first in the correct shapes and everything, and then it's replaced with bone. Most bones in the body are formed this way, but it's best observed in a long bone. Essentially, there are 6 steps for endochondral ossification. The first one is going to be development of the cartilage model. So at the site where the bone is going to form, specific chemical messages cause the cells in the mesenchyme to crowd together in the general shape of the future bone, and then develop into chondroblasts. So chondroblasts are just like osteoblasts, but chondroblasts actually produce cartilage. Because remember, chondro is actually the prefix for related to cartilage. And these chondroblasts secrete cartilage extracellular matrix, and they produce this cartilage model, as is modeled right here in number 1. So this consists of hyaline cartilage. This is the type of cartilage that the model is going to be made of. A covering called perichondrium develops around the cartilage model. Okay. The second step is going to be growth of the cartilage model. So once the chondroblasts are covered in the extracellular matrix, they are called chondrocytes. So essentially it's going to be exactly the same process as bone formation, the osteoblasts producing a lot of extracellular matrix, becoming completely covered in the matrix, and then changing into osteocytes. It's the same thing with chondroblasts. The chondroblasts will they will secrete a lot of extracellular matrix, become totally encased in that matrix, and then they are called chondrocytes. So these chondrocytes can actually continually divide and produce more extracellular matrix, and this will add to the length of the cartilage model. This type of growth, or this type of cartilage growth, is called interstitial growth or endogenous growth. So this is interstitial or endogenous growth. So this means growth from within. So this is going to be this type of growth where the chondrocytes produce more extracellular matrix, adding to the length. So this is growth from within. We're going to be adding to the length of the cartilage model. Now, in contrast, growth of the cartilage model in thickness, this is due mainly to the addition of extracellular matrix from chondroblasts that develop from the perichondrium. So this is a different process. This is called appositional. Appositional or exogenous. Exogenous growth, and this is meaning growth from the outer surface or growth at the outer surface. So this is how the model actually grows in width or in thickness. As the cartilage model grows in size, the chondrocytes in the middle hypertrophy. Now the word hypertrophy actually means increase in size. So the cells actually themselves will increase in size. Okay. So the surrounding extracellular matrix, once these chondrocytes are going through hypertrophy in the middle, the surrounding extracellular matrix actually begins to calcify. And you can see this right here, this calcified extracellular matrix, that calcification starts in the middle. The third step is going to be development of a primary ossification center. So this is important. Primary ossification proceeds inward from the external surface of bone. So primary ossification, these are going to go from the outside surface out to in. Outside surface proceeding inward. A nutrient artery will actually penetrate the perichondrium. You can see that here. And the calcifying cartilage model, it comes through a nutrient foramen in the mid-region of the cartilage model. So this actually stimulates osteoprogenitor cells in the perichondrium to differentiate into osteoblasts. Once the perichondrium starts to form bone, it's then called periosteum. So remember, this is the connective tissue that surrounds bone. Near the middle of the model, periosteal capillaries will grow into the disintegrating calcified cartilage, introducing a growth of a primary ossification center. And this is a region where bone tissue will replace most of the cartilage. Number 4 will be development of the medullary cavity, or the marrow cavity. So as the primary ossification center grows towards the ends of the bone. Osteoclasts will break down some of the newly formed spongy bone trabeculae, and this activity leaves a cavity. And this is the medullary cavity, and this is going to be in the diaphysis. So you can see that medullary cavity there in the diaphysis of the bone. Eventually, most of the wall of the diaphysis is actually replaced with compact bone. Okay, the fifth step is going to be development of secondary ossification centers. So when branches of the epiphyseal artery enter the epiphysis, secondary ossification centers actually develop. And usually this occurs around the time of birth. This is when these secondary ossification centers actually develop. So pretty similar to the primary ossification centers, though the spongy bone remains in the interior of the epiphyses, so there's no medullary cavity found here. So in contrast to primary ossification, secondary ossification actually proceeds outward from the center of the epiphysis toward the outer center of the bone. So this is when we're starting in and we're going outward. So this is going to be inward growth in the primary ossification center and outward growth in the secondary ossification center. Last step in endochondral ossification is going to be formation of the articular cartilage and the epiphyseal plate. Remember, the epiphyseal plate is the growth plate. The hyaline cartilage that covers the epiphyses actually becomes the articular cartilage, which is kind of cool. Prior to adulthood, the hyaline cartilage remains between the diaphysis and the epiphysis as the epiphyseal growth plate. So that's the hyaline cartilage that was still there during growth, and that's going to turn into the epiphyseal growth plate. And remember, this is the region that's responsible for the lengthwise growth of long bones. And remember that that growth stops between like 14 and 24 years old. Okay, so the growth in length of long bones involves the following two major events. The first one is that we're going to have interstitial growth. So we're going to have interstitial growth of cartilage. And we're going to have interstitial growth of cartilage on the epiphyseal side of the epiphyseal plate. So here on the bottom of this diagram, is where we're going to have the epiphyseal side of the epiphyseal plate. This is histology of the epiphyseal plate. On the bottom is the epiphyseal side, and on the top is the diaphyseal side. So we're going to have growth here on the epiphyseal side of that growth plate. So that's going to be the first way that we're going to have growth, interstitial growth there. And then the second step is going to be replacement of cartilage on the diaphyseal side of the plate. And this is done by endochondral ossification. So then we're going to have replacement of the cartilage by bone. And that's going to be on the diaphyseal side. So this is going to be on the epiphyseal side, and this is going to be on the diaphyseal side. So remember that interstitial growth involves continual cell division of chondrocytes and then further secretion of the cartilage extracellular matrix. So on this radiograph here of the epiphyseal growth plate, you can actually see that it appears as a dark band between calcified areas on the X-ray. You can kind of see here that dark band here. This is actually going to be the growth plate. It's that dark band. And this is because bone looks white, but anything else, like cartilage, will actually appear black. And so here on the bottom, we actually have the zones in the epiphyseal plate. There's going to be the resting cartilage on the epiphyseal side. And these do not contribute to bone growth at all. Rather, they just anchor the epiphyseal plate to the epiphysis of the bone. So next is the proliferating cartilage zone. And these include chondrocytes that undergo the interstitial growth as they divide and secrete extracellular matrix. The zone of hypertrophic cartilage will be next. And these consist of large maturing chondrocytes that are arranged in columns. So you can kind of see these columns here that are part of the hypertrophic cartilage. Next, we have the zone of calcified cartilage. These consist mainly of dead chondrocytes because the extracellular matrix around them has calcified. The osteoclasts dissolve the calcified cartilage, and the osteoblasts and capillaries from the diaphysis will actually invade the area and replace the calcified cartilage by endochondral ossification, like we covered in the last slide. So bone grows in this way by the proliferation of chondrocytes, which then die and then are replaced by osteocytes. You can see here. So this is how they grow in length in the epiphyseal plates. So up till now, we've been talking mainly about a typical long bone up to this point. Later, we're going to discuss other bone types, but we'll first finish talking about long bones. So as new bone is deposited on the outer surface of bone by osteoblasts, the bone tissue lining the medullary cavity is destroyed by osteoclasts in the endosteum. So at the bone surface, we have periosteal cells that will differentiate into osteoblasts, which secrete the collagen fiber and other organic molecules that form bone extracellular matrix. The osteoblasts become surrounded by extracellular matrix and then develop into osteocytes, just like we've discussed before. So this process forms bone ridges on either side of a periosteal blood vessel. So here we have these periosteal ridges. This is going to be creating width or thickness in the bone. So eventually, these ridges will actually come together and fuse together, as you can see here. And the groove actually becomes a tunnel that encloses the blood vessel. The former periosteum, as you can see here, going to be the outer lining, actually then becomes the endosteum, or the inner lining that lines the tunnel. Osteoblasts in the endosteum build new concentric lamellae inward. You can see that here. And this is creating, essentially creates a thinner tunnel, but it creates more thickness and it forms a new osteon. So this repeats as need be to increase the thickness of the bone. So here you can see another osteon is being formed right here. Continues and continues, forming a thicker bone. All right, here is a playable animation on growth and remodeling. Would be a good one to watch. and one on bone dynamics and tissue, another playable animation. And that is going to be the end of Chapter 6, Part 3.